Good morning, students. This is your teacher Neelam Elawat, and today we'll continue with the same chapter, that is molecular basis of inheritance. Although the chapter is same, dear students, but we will have a very innovative topic, very interesting topic, that is human genome project, right? This is a very important one. So far we have discussed about what we have discussed, students? Yes. So far we have been discussing about the various mechanisms related to the heredity, like the transcription, translation, right, replication, and then we talked about the genetic code. Yes. So these are very interesting topics and very important from your exam point of view, and this topic that we are doing is also very important one. So let's start with the today's topic, that is your human genome project. So human genome project, when initially was started, okay, so this scientific project, it deals with the study of base sequences of DNA molecules. See, we know that the DNA is made up of a double helical structure, right? And this project deals, it involves the study of the base sequences of DNA molecules in your entire chromosome set. That is why we call this project as human genome project. Means this is a project which will study all chromosomes of the human beings, will identify the different genes into it, right? So that is why it is called a human genome project, right? And it is rapidly associated with the new areas of biology, right? Clear? And the different goals of the human genome project are: first, it will help us to identify approximately 20 to 25,000 genes in the human DNA. Okay, it will help us to identify these many genes in the human DNA. Secondly, it will store this information in the database. Okay, in your computers there will be a specific database where this information will be stored. Thirdly, it will improve the tools for the data analysis. Means if you have to compare the DNA of two different individuals, you will have a sufficient amount of technology, information, so that it can easily compare. Then it transfers the related technologies to other sectors such as industries. And the most important, it will address the ethical, legal and the social issues that may arise from the project. See, students, whenever it's a very ethical issue, that no industry and no technology and none of the things that they are allowed to experiment with humans, because we never know that experiment might lead to a very disastrous effects. So that is why this human genome project, it addresses the ethical and the legal and the social issues which are related with this project. What are the different methodologies that has been used in this one? First of all, to identify the different genes that are expressed as RNA, they are referred to as expressed sequence tags. Okay, what do we call them as? Expressed sequence tags, right? So when we sequence the whole set of the genome that is present in our coding and non-coding sequences, I hope you remember what are the coding sequences. Yes, the exons are the coding sequences and introns are your non-coding sequences. So when the mRNA is formed, then your introns are removed and then only the mRNA is converted into functional mRNA, right? So when you are doing the sequencing, though all of the genome will be sequenced, which will contain both the coding and the non-coding sequences, right? And later the different regions in the sequence will be given different functions, which is called sequence annotation, right? Which we call it as sequence annotation, right? Then the total DNA from a cell will be isolated. What will be the procedure? First of all, the total DNA from the cell will be isolated and then it will be converted into fragments. Okay, now suppose this is a cell, right? This is the nucleus. In the nucleus a complete DNA is present. So all of this DNA will be removed and then it will be broken into small fragments, okay, a very small sized. So small fragments, and these small fragments then they will be transferred into the suitable host using vectors, okay. and you can say the cloning of these specific sequences will take place, which we call as amplification, right? So this small sequence of the DNA will be inserted into the host by using special vectors, and then this particular sequence will be cloned and amplified, right? Okay. The commonly used vectors are BAC and YAC. BAC means the bacterial artificial chromosome. As I said, that these small segments of DNA, they will be introduced into the special host through using the vectors. So vectors that we'll be using is a BAC, that is your bacterial artificial chromosome. What do we call it as? Bacterial artificial chromosome. And next is yeast artificial chromosome. So these two will be utilized as vectors. Then these fragments, they will be sequenced using the automated DNA sequencers, right? We will be using the DNA sequencers, and we will that is called automatic DNA sequence, and these fragments, their sequence will be determined, like A, T and GC, what is a particular sequence in these small fragments of DNA. Then specialized computer-based programs were developed for the alignment of sequence, right? So when we are sequencing this particular DNA, this particular portion, suppose this is a small fragment A, T, G, C, isn't it? We are determining the sequences. So these sequences, they will be determined by using specialized computer-based programs. Okay, and these sequences then will subsequently they will be annotated and will be assigned into each chromosome. So you can say inside each chromosome what are the different DNA, what are the different genes that are present, and what is the function of each gene, that will be determined using these specialized techniques, right? The cloning resulted into a amplification of each piece of DNA. As I said, that nucleus contains your DNA, then this DNA will be converted into small fragments, and each fragment will be cloned and it will be amplified. Okay, and then a particular sequence will be determined, right? The sequence of each will be determined, and the vectors that we'll be using is BAC and YAC, right everyone? And the sequence of chromosome 1 was completed in May 2006. This was the last chromosome that was determined. Okay, last chromosome was your chromosome number 1, and it was determined in May 2006. So just you can imagine that it was completed, the sequencing of the human genome was almost completed in May 2006. Okay, and the chromosome number 1 was the last to be sequenced. This is a automated DNA sequence. See, this is a machine which is used for DNA sequencing. This is automated. You can easily see a screen. It has a computer fitted designs, computer fitted softwares. So when you feed in the DNA into it, automatically it determines the sequence of that particular DNA, the sequence of the small fragments of the DNA, right? So what are the salient features of the human genome? Now, when we conducted this experiment, we launched this project, then what were the outcomes? What we came to know about it? It led to the development of, or you can say it is considered to be the very important project, and lead to the discovery of large number of facts. The very first important fact is that the human genome contains 3,164.7 million nucleotide bases. Isn't it very drastic? These much bases are present in human genome. So entire human genome, that is your 23 pair of chromosomes, they were sequenced, and the total was the 3,164.7 million nucleotide bases were present. And on an average, there are 300 base pairs, with the largest human gene being dystrophin. Okay? On an average, at an average, or can say if we take out the average, a gene basically contains 3,000 bases. Okay, a gene basically contains 3,000 bases, right? And the largest gene is a dystrophin. It has 2.4 billion bases. It has 2.4 million bases. Let's see, we can easily see this is a gene. This is a chromosome, and this particular portion is referred to as dystrophin gene. Okay, and this dystrophin gene is the largest known human gene, right? It is the largest known human gene because it contains how many? 2.4 million bases. So the total number of genes that have been estimated is 30,000. So total number of genes that are present on the chromosomes is approximately 30,000, and 9% of nucleotide base pairs are exactly same in all people. See, we know that we human beings, we are entirely different from each other, but through this sequences it has been identified that out of 100%, 9% of our base pairs is exactly same. So 9 base pairs are exactly same in all human beings. And whatever genes that have been discovered, there are 50% of the genes, the function of 50% genes is not known. So far, we are not able to determine the function of 50% of the genes, and 2% of the genes code for proteins. Just imagine, 9% is same for everybody, isn't it? 2% they code for proteins, and 50% of the genes we do not know their function. So far, we are unable to determine their function. And the repeated sequences make a large portion of the human genome. Means a majority of the human genome contains repeated sequences, right? And these repeated sequences are parts of the DNA sequences that are repeated many times. Okay, these repetitive sequences are the ones which are repeated number of times. Okay, chromosome number 1 has the most genes. The chromosome number 1 contains 2,968 genes, while the Y chromosome has only 231 genes. So largest number of genes are present on the chromosome number 1, and very few genes are present on the chromosome number Y, that is only 231, right? And the people who are working on the Human Genome Project, they have identified 1.4 million locations, 1.4 million places where a single DNA difference occurs. Just imagine, 9% of our genome is same in everybody, right? We all human beings have 9% of the genes are same, and 1.4 million locations are such where we only differ from each other by a single base pair. Just imagine this, and we call it as single nucleotide polymorphism. What do we call it as? Single nucleotide polymorphism, right everyone? This is, see, just an example I've quoted here for you. This is ATGC80. Again, this is the same, this is same. There are 3 persons, right? These are 3 persons, and I've taken the sequencing of these 3 persons. Just imagine, they only differ in one base pair. Rest of the nucleotide base pairs, they are same, they only differ in one base pair, only one base, nucleotide base, and that is why we call it as single nucleotide polymorphism. What do we call it as? Single nucleotide polymorphism. So we can imagine that even a single difference in the bases accounts for a lot of difference, isn't it? Even a single change will account for a lot of difference. So, right? So let's see the single nucleotide polymorphism, right? Then comes the applications of Human Genome Project. Why do you think that Human Genome Project is important? Do you think why do we sequenced all the 23 pair of chromosomes? Is it important for us? Just imagine, what was the need? Why we wanted that all the genes we should know which chromosome contain which genes? Why we wanted to know the functions of genes? Is it important? Yes, it is important, because this will help us to study all the genes that are present in the genome. And secondly, it will help us to understand how particular gene works and how protein works, how their functions are interconnected. And the very most important one, that is, it will help us to understand the various genetic diseases and to establish a cure of those diseases, isn't it? Right now we know that it's a trend. We are sitting in our houses because of the coronavirus. So if we are able to manipulate the DNA of the coronavirus, if we are able to play with DNA of the virus, then 100% we will be able to establish some vaccine for this. So studying the genome is very, very important, students. Right? Studying the genome is very, very important. And if you remember, we have talked about hemophilia, isn't it? The different genetic Mendelian disorders. So even a difference in a single base pair can account for a disease. So this will really help us to understand the various genetic diseases, and we will be able to establish a cure or to find out a particular cure for those diseases. Right, everyone? Now we understand that why we have trisomy, isn't it? I hope you understand why we have Down syndrome, right? Why we have Klinefelter syndrome. So this is because we now know these sequences of our genome. So that is very, very important. So last point is the most important point, that it really helps us to diagnose, to know why a particular disease is being caused and what is the treatment for that particular disease. Right, everyone? Okay, so coming to your homework for today, first of all, to make notes of this topic, then you read the page numbers 118 to 120. Your complete human genome project is described in these 3 pages. You must go through it, underline the important points, right? You must go through it and underline the important points. Right, everyone? Okay, that's all from my side. Bye, everyone.